The most common property of all living organisms or all propagating entities is that they all have genetic information. In propagating entities could be cellular forms and acellular forms. Cellular forms include bacteria, eukaryotes, including starting from protozoans, fungi, plants, animals, and humans too. Acellular life forms include, or acellular propagating entities include phages or viruses, plasmids, and viroids. In the cellular life forms, double-stranded DNA is the genetic material, while in the acellular forms it could vary. Some of the viruses may have single-stranded or double-stranded RNA, and some may have single-stranded or double-stranded DNA. So, at least in cases of the propagation-wise, all life forms depend on cytoplasm in a way. That means even the acellular life forms have to propagate only in the cellular life forms as parasites. And how does the genetic information of the cellular life forms, how does it manage these things? How does it maintain it in a physical form? That is what we will discuss in this series of small talks. The cells are small, and DNA is a supercoiled DNA, and it is actually like a wire, very long. And then it is packaged into the cells, and there is one of the important parts of it, or the points that we should remember, is that the cell itself is the physical manifestation of the genetic information it has. And the second point is the cell has to give rise to new cells, such as these daughter cells here. And then those daughter cells should have genetic information so that they will manifest their physical form. Not just the physical form, but also how they should develop, how they should respond to the environmental stimuli. And that is how multicellular organisms will develop into a complete organism and work together to eventually make more copies of themselves. So how does this happen is an interesting thing. That means the genome should be, while it should express itself, it should also be protected, because it is vulnerable to damage or mutations. And at the same time, it should be protected, but at the same time it should also allow the expression of those genes so that the cell can amass more material and try to propagate. So what we have in this is that all the DNA in all the cells is wound around several proteins. This is much more characteristic in terms of eukaryotes, because the DNA of eukaryotes is wound around special proteins, basic proteins, or proteins with a lot of basic amino acids called as histones. They are basic in nature because they have large amounts of arginine and lysine on their surface at least. And they should be basic because if they are to bind to DNA, and DNA is acidic, then they should have basic residues so that they can interact together strongly. So DNA is wound around these histones. Even in bacteria, there are some proteins. It's not as compact as we see it in eukaryotes, but it is still, all chromatin has proteins as well as histones or histone-like proteins. Not only these, there are also many other proteins that bind, which are regulatory in nature, which are involved in regulation of several genes, repression included. So if you look into the cells that we have about 3 × 10⁹ base pairs in a haploid set. That means we have two, we are diploid, which would mean we have 2n. So that means we have about 6 × 10⁹ base pairs. If we see it this way, and we have 3 × 10⁹ base pairs, and each of the base pairs occupies about 3.4 angstroms. If you remember that one turn in a in a B form of DNA has about 10 to the power 10.5 base pairs, and approximately we will take it as 10 base pairs. And the length of the height of each turn is about 3.4 nanometers or 34 angstroms. And if we divide it by 10, we get 3.4 angstroms. And then if we multiply these number of base pairs with that of 3.4 angstroms, we get 10 to the power 10 angstroms, which is nothing but 1 meter. And we have diploid set. That means the total length of DNA from one cell, if all the chromosomes are put end to end, we get about 2 meters. That is longer than or the taller than height of an average human. Average male around the globe, of course, is about 1.7 meters. And the DNA in one cell is longer than that. Now, if you imagine the body has about 37 trillion cells, and imagine if we put all the DNA end to end together like this, it will be interesting if you can calculate how many kilometers it would make. And similarly, we have the length of DNA is about 2 meters, and the size of the mitochondria is about 10 micrometers. How does it fit in? And that is by binding with the histones in the form of nucleosomes. Let's calculate compaction. The size is about 2 meters, and we have about 2 into 10 to the power 6 micrometers of DNA. The length is about 2 into 10 to the power 6 micrometers. I just converted meters to micrometers. And the size of the nucleus is about 10 micrometers, say approximately. And we divided with 10 micrometers, we get 10,000 fold. The compaction is about 10,000 fold. That's very impressive compaction. You can imagine 2 meters of DNA and all of it coiled upon itself into a very something that we cannot see with our naked eye. And that is how the nature solves the problems. And it'll be, as we go forward, we will also look into more of these things. Thank you very much.